Okay, so today I'd like to give a little bit of a discussion on some of the concepts we've developed for network science and looking at embryos using network science, particularly two related concepts: the density bifurcation model and the generative divergent integration concept. So let's start with the density bifurcation model. So as we're aware, if we have, say, a single cell embryo, that cell will divide into two and divide into four and divide into 8, and we end up with these divisions that form. So we end up with something like this. So that's a lineage tree, and it's a binary tree in the sense that it divides into two daughters. Now, this is excluding asymmetric divisions, but for now, we don't care about that. So we end up with these cells, and once they start to divide, then they start to acquire some function. So we have these 8 cells, and at this stage, they maybe don't develop a function. But let's suppose that they maybe start to develop a function. If we put 8 cells in a circle like this, maybe 3 of these cells are destined to become one type of function, and the other cells are destined to become some other type of function. So we have 2 sort of subsets of this structure. Now, we could just say that those 2 things will divide and break away and call it a day and say, okay, we can take our lineage tree and we can say, it's sort of segregated off into some area. Like we can take these 3 cells here, say those are a separate module, and then the rest of these cells here, these 5, and those are a separate module. But you notice 2 things. One is that this set of cells are in 2 different groups, but they share a mother cell. So sometimes daughter cells will inherit things in the cytoplasm from mother cells. Sometimes they're part of the same sublineage, and so forth. The other thing is that these cells weren't going to just stay sort of on their own sides. Sometimes they'll start to integrate functionally with cells in other groups. So if we have something like different organs that develop, like if we have a nervous system start to develop, or a connectome, and then we have, maybe something like a germline down here, and an intestine, like we have in C. elegans. And all these cells, they're going to connect with each other in different ways, because actually maybe not so much the germline, but you get different connections, functional connections, structural connections. So these cells don't remain in these modules, and they don't remain separate things, at least not in the same organism. The germline might be an exception because it usually gets segregated out from the rest of the lineage tree early, but nevertheless, it's part of the same organism, and so it's always going to have some interaction with these other cells. So what do we do about this? It seems like we could build a network and we could just start to proclaim modules, and we could start to proclaim this sort of structure and be done with it. So the first idea here is the density bifurcation model, and this is something that we've developed back in 2022 and presented this at NetSci 2022. So the density bifurcation model basically allows you to have increased connectivity as you start to increase the number of cells in your embryo, actually in development more generally. And so, this increasing connectivity is something like what I showed up above here, where we have these different types of cells, and, we can draw connections between them, which are maybe interactions, or maybe they're physical connections, or maybe they're chemical connections, something that has to do with them interacting or being a part of the same thing, although not necessarily maybe being from the same part of the lineage tree. So this is basically, this is kind of a rule that as we increase the number of cells in development, we get increased connectivity. All right. So we know that cells divide and migrate. So they not only divide, but they also migrate. And so you have two things here. You have more cells, but you also have, the cells that sort, cells that move around, cells that maybe had one neighbor at one time and another neighbor at another time. And basically, this is the process we need to go through to get organs and tissues. And so you see that this sort of organization isn't just simply separated modules, it's actually this sort of integration where cells are within the same sort of sphere of influence and they're moving around and interacting in different ways. And we need a model to describe this. So the idea is that you have, as you get more cells, you get this bifurcation of function, but you also get this interreliance or interdependence. And so we can represent this in a network just as well as we can represent it by just verbally. Another thing about this, we started with this main idea here. We have our first point. Our second point is that cell migration enriches local communities and cliques. What do I mean by that? So what I mean by that is that we have these structures in networks, these little communities that are sort of, connected things. Sometimes we do community detection and we do the algorithms for that. Sometimes we have cliques that are very densely connected groups of cells, and we can also look for cliques using algorithms. But we can actually, in this case, connect developmental biology and network science by defining these sort of spatially organized structures. Instead of just, say, looking at a lineage tree and saying that there's a sublineage, we can actually look at these cells that are organized in space, and they might be part of the same tissue, but they might also share this sort of developmental sort of sphere of influence that can be defined by communities and cliques. So this is we'll call it a sphere of developmental influence. And so this isn't just simply like finding static cliques and static communities either. This is actually something that where the cells migrate around, and maybe there are some discontinuities at least at any one time point. So over time, these cells interact, but you don't necessarily see that signal at any one time point. Nevertheless, they still have connections that we can characterize. So this requires a dynamic network where you have a network structure that relies on the evolution of the connections between the different cells. And so this is one of the many things that's led us from, sort of traditional classic network science to looking at hyper networks and hypergraphs and graph neural networks as a means to look at this. And so we have this other set of conditions, which is that we have these cells and they're growing in size and they're migrating around and they're making these interactions and they're forming these communities and cliques, and sometimes over time, sometimes discontinuous. But eventually they'll start to diverge in terms of their differentiation. So the cells themselves will differentiate, again, like we showed before, into these different functional classes. We might have like an intestinal cell and a neuron and a muscle cell. And these are going to be distinct identities for these cells. And many cells will do this at once. So they form a tissue, something like muscle, where you have a bunch of muscle cells, or something like an intestine, where you have a bunch of intestinal cells. It doesn't mean that they don't interact with one another. It just means, or maybe they're from the same lineage. It just means that they are separate functionally. So they'll tend to be together. They'll tend to be connected in different ways, spatially, with spatial proximity, chemical signaling, whatever. On the other hand, they also still have this sphere of influence between one another. They might share a common origin in a sublineage, or they might be, next to one another earlier in development, and thus share some connection earlier. So on the one hand, you have these things that are segregated in the later embryo, maybe sort of nearby or in proximity in the early embryo. And so they might form one of these communities but belong to other tissues. And this is what we call sort of this bifurcation, which is where you get this bifurcation, which is this division of labor and structure with maybe what we'll call weak ties. So if we remember our network science, weak ties goes back to a lot of social network work in the 60s, where they're looking at social networks where you have individuals that sit between different groups and they serve as this weak tie. In other words, they're never highly connected to either group, but they serve as a conduit between the groups. And so this is, where we have maybe these tissue types. We might have cells that connect these tissue types. They might be intermediate cells. They might be like connective tissue, cells that belong to connective tissue, or cells that are relay cells that might relay a message between, say, neurons and muscle. There are a lot of potential candidates for this. But the idea is that these tissues, as they separate out spatially and functionally, are connected through these weak ties. So this is where we get this basic connection, and I'll talk about this in terms of generative divergent integration in a minute. So this brings us to our fourth point, which is these interconnected networks provide us with a candidate list of weak ties. So we have and so weak ties are interesting because in a lot of ways they serve as sort of these conduits between different categories. They're not like if you've heard of rich club neurons in the connectome of C. elegans or other connectomes. This is not exactly like a rich club. A rich club is highly connected. It's a hub. It's densely connected, but has influence with other places. A weak tie will be generally very weakly connected, but link maybe two very large networks. And so they share this weak tie, which is to say that if you cut the tie, so we have these two very large networks here, and I'm just going to show this very briefly. If you cut the tie here, you cut this weak tie off to the other network, then it really becomes useless. It just becomes a terminal connection, and it doesn't really have any consequence. If you, maybe were to rejoin the connection here, you would then be able to reestablish a connection between these two networks. And you might ask, why don't these networks tie directly? And the answer is that they very much can. But when you have this kind of connection, these cells in here in each network serve as the weak ties. And if you cut this connection here, then these cells no longer have that weak tie role, but these networks, of course, then are severed. So there are a lot of ways these weak ties sort of play a role in integrating the overall anatomy of the organism as development proceeds. You get these tissues that sort of, they bifurcate in terms of function, in terms of structure, but you still have these things in the middle that provide functional integration. So that brings us to our next point, which is generative divergent integration. And this is a concept we talked about in a set of, we talked about this in the 2021 talk, and we've kind of developed this idea over several years at a number of network science conferences, and I think it's included in our archive preprint on hypergraphs and development that we have from this last year. So we do kind of touch on that topic. We really do need to write up a preprint on this specifically because I think it's a very important concept. So generative divergent integration is basically this idea that as you increase the number of cells, you get these networks that start to form as the cells differentiate, and you get these independent sort of spatial and functional modules. You get these weak ties between them, but you still get this generative divergent integration. So in other words, as you're increasing the number of cells, the chances of getting different cell types increases. And so this, sometimes this is because the cells are programmed to differentiate at a certain point in development. Other times it depends on what the sort of the microenvironment of the cell is. So if you have a greater number of cells, you might get a multitude of signals and you might start to get these different tissues form. So we'll say A is increasing number of cells, chances of different cell types increases. B, you get an increase in the number of cells in your embryo network. Increases in size. But also sort of in the number of opportunities for these weak ties. Actually, we don't really get the weak ties yet. We get sort of this opportunity for new communities. As you remember, we have a lot of migration and division and differentiation. Okay. Then in C, when we get our new communities, the new communities will tend to be segregated in terms of space, so spatial and functional. Okay, so then that's C. And then D, we have, as we get these different spatial and functional segregation points, then we get these weak ties that form. And then we get our, integration. And so C then is this idea that there's this generative process. Actually, it's A through C. Because remember, we're increasing the number of cells. We're getting these networks that form. You're getting migration. You're getting differentiation, and that's forming these modules or these communities. And then you're getting these new sort of tissues that come out of that. So it's all generative process, multi-step generative process, but still. So that's pretty much it. So let me share a couple of slides with you to kind of drive this home. So the first example is of this sort of generative divergent integration model is that you get the formation of these anastomoses. So what you get here is you get these different parts of a network. They might form different parts of a tissue, different layers of a tissue, different tissues themselves, different cell types, and they're all connected. And then you get these like these connections between the subgraphs, which are weak ties. This cell and this cell are weak ties. This cell and this cell is a weak tie, and you get these connections between them, bridging them, if you will, forming what we call anastomoses. And in anatomy, an anastomosis is like something like a sphincter or something like a valve in a heart that is a cross connection between, if we represent a heart as a subgraph or a gut, a stomach as a subgraph, we can get these structures that surface, these weak ties or anastomoses. And so this is where we're starting to get this kind of structure. And you see this a lot in very early development, even with neural tube closure and things like that. So that's one example. The other example is this example of generative divergent integration. And this is kind of an example from one of our talks where we have relatively few cells and we get these discontinuous networks that don't really do anything. They're just kind of like, based on our limited criterion of connectivity, we get these, these disconnected networks. But then as we go and we get more cells and they move around, we get this 24 cell example where we start to get these densely connected subgraphs. And then, they start to move around and you start to also get these weak ties between these functional subgraphs. And so this 8-cell example shows something that isn't functionally divergent. There are these sort of discontinuous pieces, cells that are connected, or discontinuous subgraphs. I wouldn't even call it a subgraph, but basically that doesn't mean anything because it's not functional and it isn't really very highly structured. In the 24-cell example, you start to get a high level of structure, a high degree of centrality. You start to get some, some clustering in different parts of the embryo. If you look at this gray oval, which is the embryo, and so you get the opportunity for weak ties, as well as these highly dense, highly dense communities. And so this, again, with embryo networks, you have to recall that the connection criterion can be anything. It can be spatial distance, it can be chemical signaling, it can be physical connections like gap junctions, or chemical connections like synapses. So we can use this in a number of ways, but these rules should hold true across all these different criterion for connectivity.